This video is going to overview plant life cycles, which is something which my general biology students have struggled with in the past. And I think the reason for that is that when we think of our own human existence, or even that of all animals, that the diploid phase, well, that makes up the whole organism. So the multicellular part of our life cycle is diploid, having two copies of each chromosome. The only part of our life cycle where we have haploid cells are the one cell stages around reproduction. So sperm, in the case of humans, have 23 chromosomes, one of each kind. And ova have 23 chromosomes, one of each kind. But all other cells have two sets of these 23 chromosomes for a total of 46. This is the diploid count. And so when we think of haploid cells, we think of the gametes, which exist more or less simply as a single-celled individual, whereas here the multicellular part of our life cycle involves cells which are diploid. So liver cells, or liver cells, actually a bad example. Skin cells, bone cells, smooth muscle cells, nerve cells, etc. They are diploid with two copies of each chromosome. Plants are different, and because they're different, it's a little harder for us to understand. And so that's why this lecture, I think in the past has given some students a bit of trouble. Let's begin with the green algae, simply because it is from green algae that plants evolved. And green algae like Spirogyra here typify the case of the green algae. These are the photosynthetic strands. So when we think of the alga, it's colonial. And so we're probably thinking of these cells. These are haploid. There is one set of each chromosome. However, when it's time to reproduce, neighboring strands can develop a conjugation tube, which allows the cytoplasm and nucleus of one cell to then enter the cell to which it is now fused. And this then forms this single diploid cell. All right. So these cells are now sexually reproducing. Now, they didn't have sperm and ova. They didn't have specialized haploid cells. Any haploid cells could fuse through a conjugation tube with the haploid cells of the neighboring strand to make now this diploid cell. Now, this diploid cell might then last the winter. Because very often in freshwater environments, there is rapid growth in spring and summer, but then with the onset of the colder weather, there's more of a dormant phase. So these diploid cells, they may last the winter, but when it comes time to develop into photosynthetic strands, it is not the diploid cell that does that. Instead, the diploid cell now undergoes meiosis again. And now the daughter cells of meiosis are haploid cells, which then develop into new strands. And so it is the haploid phase of the life cycle, which is the dominant phase and is the vegetative phase, which is doing photosynthesis. The diploid phase exists as this zygospore, this one-celled celled stage. Now, I would like to get into plants. Green algae are obviously protists. There are different types of protists and different types of algae. And some, for example, the red algae and the brown algae, they are far more complex in their reproductive structures than that. So, for example, if we were to look at the brown alga fucus, unlike what you saw there are multicellular structures involved in reproduction. So this is the oogonia, which is making the haploid ova, and then there are spermatogonia, which will make the sperm. And so while green algae can be very simple in their reproductive structures, there are different kinds of algae which aren't that closely related, and that both brown algae and red algae can be more complex than the green algae. But since plants evolved from green algae, let's then go back to plants once again. So when the algae from which plants evolved have the haploid phase as the dominant vegetative part of their life cycle, and that the diploid phase is short-lived, just around reproduction. And if we were to look at the most primitive plants alive today, the liverworts, we see just that. So here is the plant, okay? This is the part that's doing photosynthesis, but this is haploid. So just like the alga, the vegetative part, the part that's doing photosynthesis, that was the haploid part. The same too applies to these primitive plants. Now, it's hard for us to relate to that. It's as if almost that haploid human cells have grown up into their own people, which obviously they don't. In us, the haploid stage is just a single stage. Now, on these liverworts, however, then reproductive structures can develop, two different kinds. So the same plant can develop both antheridia, which will develop as sperm, and archegonia, which will develop ova. So here is a picture of antheridia, and those are not only sperm, but they are flagellated sperm. They can swim. This is one of the reasons that the primitive plants are so dependent on water. And they are trying to swim up this tube of the archegonia, where the ovum is being produced. And so these haploid masses of vegetative tissue produce haploid sperm with flagella, which can swim, and then haploid ova. After fertilization, we then get to this phase, which is now diploid. That was called the sporophyte. Okay, now I'll show you images of all of those in a second, but just there are two things that we just saw which make the plants more advanced than the algae with regards to reproduction. There would be others, for example, a cuticle, a waxy cuticle, which protects them from water loss. But notice that here, the diploid phase, it's not very big, but it is multicellular. And that does distinguish these liverworts from the algae. Also, if you look, the gametangia, the structures which are producing the sperm and the ova, are multicellular. The green algae don't have that. And so because the structures producing the sperm and ova are multicellular, and because the diploid phase is multicellular, there have been some advances since the algae. One final thing is that there are different ways that plants can reproduce asexually. One of the ways is that these liverworts can produce these cup-like structures known as gemmi, which then just have little masses of cells which have not been produced through sexual reproduction. But nevertheless, those, say in a rain, could be washed somewhere else and then germinate to become a new plant. Now, in general biology, too, students often have to then identify structures under the microscope, so I'll focus on that. So here are antheridia of liverworts. It is a stalked structure. Once again, all plants, including these primitive liverworts, have multicellular gametangia, and it's here where masses of sperm are produced, and they do have flagella and can swim. One of the reasons that the reproduction of liverworts is tied to water. The archegonia is produced in a structure, it's a structure which produces the ova and includes a tube through which the sperm will swim. Now notice that just as in animals, sperm are cheaper to make, so a greater quantity is made, a fewer quantity of ova are made. And then also notice here in the archegonia, there are very large cells which are the ova which have the nutrients to begin then the next generation. And so the Archegonia are producing fewer gametes than the Antheridia, which produced the sperm, which they did. So notice there's a tube through which then the sperm can actually swim. So you can see the lumen of the tube. The sperm can swim to reach the next slides will be of the sporophyte stage. And so this is dependent on the gametophyte. The gametophyte is the haploid phase. And the sporophyte phase is not free living. It's going to then simply grow in the place where the Archegonia was fertilized. So once sperm fertilized the Archegonia, it then develops into that multicellular sporophyte structure, which will then undergo meiosis and produce haploid spores, which will then become the new haploid gametophyte generation. So it's multicellular, once again, more advanced than an alga. But it does not perform photosynthesis. It simply exists for reproduction. Okay, so that was the sporophyte stage, and I think Lycopods are interesting because obviously in the mosses and the liverworts, the gametophyte and the sporophyte are not equally represented in the life cycle. The gametophyte dominates. In all of the next groups, the sporophyte will dominate. But here in lycopods, you actually find a condition where it's closer to equally represented. Interestingly, the gametophyte is often a subterranean structure. It is free living, so neither generation depends on the other. The gametophyte is free living, the sporophyte is free living. The gametophyte can live a number of years, even more than a decade. And so even though when we think of lycopods like club mosses, we think of the green part of the life cycle, which is photosynthetic, there is a whole separate other phase of the life cycle, which lives years, but can be decomposing underground. And so the haploid spore germinates into the haploid gametophyte, once again, could be underground. It's that gametophyte that produces Antheridia and Archegonia. And from that, then we get the diploid green portion of the club moss, which is free living and photosynthetic. Here, this portion then produces spores after meiosis occurs, and then the cycle can begin again. So I'm going to focus more on ferns, which students have a bit more familiarity with. But once again, as we look at the balance between the gametophyte and the sporophyte, lycopods are interesting in that you could argue that it's close to the two representing equal portions of the life cycle. In ferns, the haploid spore does grow into a haploid free-living individual, which is green and performs photosynthesis. It, however, is really small, and it is not the dominant phase of the life cycle. So even though the gametophyte and sporophyte are both free-living, the gametophyte is a minor portion of the life cycle. However, it does produce the gametangia, so the Antheridia, which produce haploid sperm, which still swim, given their flagellar tails, and ova from Archegonia. The sperm can then fertilize the eggs in the Archegonia, and now that there is a diploid cell, this then grows to be the sporophyte, the diploid sporophyte, and this is the fern that we typically recognize. Although it should be pointed out, there's two stages of the life cycle. this small heart-shaped prothallus, and then the large fern, which we're more accustomed to. So the sporophyte is the dominant part of the generation, but both are free-living. The sporophyte then performs most of the photosynthesis, but then somewhere in the sporophyte, meiosis will occur to produce haploid spores. Different ferns vary in where these spores are produced, sometimes on the underside of leaves, sometimes in the middle of a leaf, sometimes in their own strobilus structure on a specialized leaf that only does reproduction. And so, oh, I'm sorry, not the strobilus, a specialized leaf that only does reproduction. And so these spores can come from different places. Okay. So if you were trying to wrap your mind around that and it still wasn't working, but that the monsters and the robots helped. So here, the haploid spores produced by the sporophyte to produce the gametophyte generation. The gametophyte generation would also then be free living, but it would be rather small. Okay, so it's a small gametophyte. The gametophyte then produces those antheridia and archegonia. All right. And then from the fertilized archegonium, now the sporophyte develops, which is the dominant phase of the the life cycle. Somewhere in the sporophyte, then meiosis would occur in a diploid sporangium, and then haploid cells would result. Okay. Now, once again, my students are looking under the microscope and identifying these. So here's the prothallus. This is a fern. Now you might say, oh, no, fern is the, so fern is the structure that I'm used to, say, growing in my garden. Well, yes, that is the sporophyte part of the fern, but here is the gametophyte. The gametophyte is very hard to see, a tiny little green structure, but here there are both the antheridia and the archegonia. Both are more circular, but we can distinguish between the antheridia because there is no tube through which the sperm pass, and you simply see, a round cluster of cells which all look about the same. So these are the sperm. When released, they will use their flagella to swim to the ovum, and this is what the antheridia of the sperm gametophyte looks like. Now, on this heart-shaped prothallus, there are also the female reproductive structures, the archegonia. And the archegonia have a large cell in the middle. That is the ovum. You can see it here where the two lobes of the heart-shaped structure meet, as opposed to the antheridia, which would kind of be near the base. So you can see there's a large, so there the antheridia now, they're near the base where the two lobes are meeting. This is the archegonia. So there's the big ovum in the center. And if you change the focus, you can see the narrow tubes through which the sperm will swim there. You can't really get both the tube and the ovum in focus at the same time. So you have to adjust the focus because, they have height to them. And so you won't see them clearly in the same image. Once again, different ferns vary in where they put their spores. So here's a specialized leaflet of a fern that only has spores, whereas on this image, the spores are located on the underside of the leaves. And so the leaves are diploid, and then we have these diploid sporangia, which and then undergo meiosis to produce haploid spores, which will then produce that heart-shaped gametophyte once again. Okay. Then when seed plants evolved, there was a switch where the gametophyte was no longer free living. So the gametophyte is going to be produced in side of the sporophyte rather than being its own separate structure. And so a haploid a spore will develop, but there are now two different kinds of haploid spores. And that's why seed plants, as I'll go through later, are called heterosporous. Hetero means different, while the haploid spores of the previous plants all look the same. They are homosporous. So a microsporangium produces microspores, a megasporangium produces megaspores, and this then develops into the male gametophyte and the female gametophyte. Now, these male and female gametophytes, they are small. So remember that prior to this, the gametophyte generation was free living. But here you have a small structure with 8 nuclei in gymnosperms, but only 3 nuclei in angiosperms if we're talking about the male gametophyte, or about 1,000 cells for the female gametophyte in gymnosperms, or 8 nuclei and 7 cells in angiosperms. So the gametophyte generation is no longer free living, but it's now growing within, say, a cone, or within a flower, or in the pollen grains, which are now reaching the female cone or the female part of the flower. So now the sporophyte is not only the dominant generation, but it is the only free living generation. So the gametophytes are dependent on the sporophyte. When the pollen grains then reach the ova, they fertilize it, and then a new diploid. So here you add the pollen. Sorry, I have trouble in going over this bar. When the pollen reached the female parts of the flower or the female cone, then the fertilization results in a new diploid organism. So let me just kind of review this in both conifers, which are gymnosperms, and then a flowering plant. Okay. So, I know a lot of people feel that they could recognize pine cones, but there are, in actuality, 2 kinds of pine cones. There are the male pine cones, and there are the female pine cones. And a lot of people, when they think of pine cones, are thinking only of the female cones. So, I think I skipped one. So gymnosperms, here the organism that we think of, the pine tree, that's the sporophyte. Obviously, note the difference between mosses and liverworts, where the individual is the gametophyte, the haploid generation. So here, the dominant photosynthetic free living part is the sporophyte generation. So there's been a complete change in the strategy of the life cycle of these plants. And the sporophyte then makes 2 types of cones. The pine cones that we normally think of, these are the female cones. They're much larger, and they may last for multiple years, as we will see. The male cones are temporary, and they are much smaller. So these are the male cones. Lots of individuals wouldn't recognize these as pine cones, but they are. They're just the male cones which are producing the pollen. They will typically only do that at a certain time of the year. You may notice that you park your car under a pine tree, and then it's covered in yellow pollen. Once again, during certain times of the year, a great amount of pollen is produced here. The pollen from these male cones then wants to be blown by wind to female cones. And so there is some difference in cones. So for example, cedars look different from pines. But here you see the female cone, and the pollen will grow into the spaces between these megaspores, pollen will grow into there, where the archegonia is going to be fertilized. And so there are these two different kinds of cones. Larger, weirder ones. These are the female cones. If you were to look at them under a microscope, you can see differences. So let me, show you both a cartoon version and then the microscope slides of them. So the small male cones we could refer to as microstrobili, or each one the microstrobilus. There are specialized leaves which will produce the pollen. These are called the microsporophylls, and this region where the pollen will be produced is the microsporangia. So the microstrobili, the male cones, make these specialized leaves, the microsporophylls, which make these microsporangia. And the pollen grain, which is the male, there are spores in this structure, and very often it develops these extensions so that the wind is more likely to take it. These pollen grains want to be blown by dumb luck and reach a female cone. And when they do, they will grow. Now, this is the haploid stage of the male. So this is the male gametophyte. In mosses and liverworts, it's a free-living plant that's doing photosynthesis. But here, it is multicellular. It doesn't have many cells. But it is multicellular and is going to form a pollen tube, which will then grow into the female archegonium and then allow for the sperm nuclei to fertilize the ovum. Now, while cycads, which are tropical gymnosperms, still have flagellated sperm, these cells are not flagellated. And so the male gametophyte does produce sperm inside the pollen grain, but these are not flagellated sperm which are swimming. They simply then grow within a pollen tube. So this is the male gametophyte. Once again, it still is multicellular, but it is small. It is no longer free living. Now, the female gametophyte uses similar terms. So the female cone is called the megastrobilus. The archegonia is part of this specialized leaf known as the megasporophyll. And then the structure here is the megasporangium, which will then produce the female gametophyte. There is an integument which covers it, all right? And then inside there is then the ovulum, the ovule and the nucellus here, which is the megasporangium. There are about 1,000 cells here, so this will become the female gametophyte. And once again, there are about 1,000 cells. So it's multicellular, but once again, it is not free living. It is inside the cone of the female cone of the pine tree. And so that is, it will not do photosynthesis, and the female gametophyte will spend its entire life there. Inside the female gametophyte, once again, about 1,000 cells. There is an archegonium that makes the egg. Now, when the pollen grain then adheres and then grows here, it passes through this space, a pore known as a micropyle. And it may take about a year for the pollen tube to then fertilize the eggs in the archegonia. And then the diploid embryos may spend another year before the seed falls to the ground. And so once again, the pollen grain fertilizing the archegonium could take about a year, and the maturation of the sporophyte embryo could take about a year. So once again, those female pine cones, they are more permanent structures, staying on the tree for about 2 years, as opposed to those more temporary male cones, which are only produced on during the part of the season where that species produces pollen, like often in the spring. And so if you were to look then under the microscope, it's easy enough to distinguish between them. So this is the male pine cone. So it is the microstrobilus. We can see the microsporangia where the pollen is being produced. And when we take a closer look, you can see that this has come from a diploid cell which has undergone meiosis, and you will see then the multiple haploid cells which then have resulted from that meiosis with these extensions which help the wind to carry them. Okay, and so lots and lots of pollen are produced then in the microsporangia on the microsporophylls, those specialized leaves. So here you can see those are the pollen grains, and you can see when I look a little closer that meiosis has then produced multiple haploid cells. Okay, so there you can see the cell division producing multiple cells here. So these are the pollen grains of the gymnosperms. The female cones look different. So here you can see the female cone. Once again, instead of lots and lots of pollen grains being produced, which have little nutrients in our small cells, there's going to be a large ovum produced. And so here you can see that specialized leaf, that's the egg there. And so the female gametophyte all holds about 1,000 cells, so bigger than the male gametophyte. And once again, it will form an archegonium with an egg to be fertilized. Right, so there's the egg. So here's the cone is called the megasporophyll. I'm sorry, the megastrobilus with specialized leaves and megasporophylls, which then produce the female gametophyte. Now that female gametophyte will have a space, a micropyle through which the pollen tubes can grow. And if you were to then look at pollen and allow it to germinate, you'll see the pollen tubes forming. Okay, there you can see a pollen tube. So here you can see pollen tubes. They will then take about a year to grow through the micropyle and then reach the eggs in the megasporangia to fertilize them. So they'll be growing through this gametophyte tissue, the female gametophyte. The female gametophyte is about 1,000 cells, once again, a bit bigger. And then after being fertilized, the sporophyte embryo will spend about a year developing into the seed, which will then be released from the plant. The last plant to consider in this set is are the angiosperms. Flowering plants are the dominant plants on Earth, but they're also seed plants. And like gymnosperms, their sporophyte generation is the dominant generation. That is free living, it is photosynthetic, and the gametophyte is even more reduced than what we saw in gymnosperms. Now, just because I'll be using these terms, let's just go through the basic structure of a typical flower, although these can vary from species to species. So the sepals are the outermost whorl of specialized leaves. Very often they are green and protective. The next layer, the corolla, has the petals. These are often colorful, but they do not have to be. They are colorful when a plant is trying to attract an animal pollinator. The third ring is known as the androecium. This possesses the stamen, and a stamen has an anther atop a filament. The filament is the stalk. The anther is where the pollen is made. And then the center structure is the gynoecium, which has the female structures. This would include the stigma, where the pollen grains land, the stalk of the structure known as the style, and then the ovary with the egg. Now, this entire structure is called a carpel, and a gynoecium may involve multiple carpels, but here we see one. Okay, so the reproductive structures in the flowering plants occur in whorls typically. Now, this could vary. I mean, some plants make separate male and female flowers. In some flowering plants are separate male and female individuals. And so there are so many diverse flowering plants. There's not one structure that they all possess. If you look then at flowers in the wild, you can see this. And so very often you see here, notice the pinker tinge. This is the gynoecium, that portion which then is going to have the stigma where the pollen grains fertilize the where the pollen grain lands. They will then grow through the style. But notice around them, these are the male structures which have the anther where the pollen is made atop a filament. Now, once again, each flower has a different structure, but the pollen grains are produced here. Should they grow then through the style, they then grow as a pollen tube, which will then go through that pore called the micropyle. Again, so there is an anther where the pollen grains are made, and these are then released and will then attempt to reach the female gynoecium. Now, remember that this is the gametophyte. Now, the gametophyte here is just consisting of the pollen grain, and notice that meiosis is occurring. You can see the division of the cells there. Now, each flower would look different. I think very commonly in lab, a lily is examined. So let's look at the lily here. Notice, remember that I said that the structures of the flower exist in whorls, with the female carpel being in the center. So this is the female carpel, and then these are the anthers making the pollen around it. We'll look at them in the next video. So here you can see the female carpel making the ovary with the egg, which will be fertilized. All right. I notice it's larger than the pollen grains, which are once again small. They will be blown perhaps by the wind or carried by an animal and grow into a pollen tube. So it's easy to identify the female ovary of the flower, as opposed to the male structures, which you'll see in the next slide. So inside those anthers, once again, the pollen grains are produced. Now, since the original tissue is diploid, then obviously produce the haploid pollen grains. Then meiosis is occurring, and so that's occurring here. In the anther, in this microsporangium, you can see the cell division, which is producing the haploid sperm. Now, they are no longer flagellated like in the majority of the gymnosperms, so they do not swim to the ovum. They now have to grow in a pollen tube, so that the male gametophyte, once again, is the pollen grain and the pollen tube. So notice the cell division going here to make those haploid to make those haploid cells. Okay. Now, angiosperms have an interesting type of fertilization called double fertilization, which makes them unique. So if we were to ask here, how many cells compose the female gametophyte? Well, the answer is, after some cell division, we end up with here the egg, which is to be fertilized. So the sperm will develop here through the micropyle to reach the egg. Neighboring the egg are two cells called synergids, At the opposite side are 3 cells called antipodals. And then there is one cell that has 2 nuclei called the polar nuclei. And so if you were to ask how big is the female gametophyte, well, it has 8 nuclei, but 7 cells, because here the polar nuclei, they exist together in one cell. When the pollen grain, when the pollen tube forms, so here you can see the pollen tube growing from the stigma where the pollen germinated, through the style, through a pore called a micropyle, then 2 nuclei are going to be important because this will be a double fertilization. One haploid nucleus will fertilize the egg, but one haploid nucleus will then fertilize the cell that has the 2 polar nuclei and form what's called the triploid endosperm. So there was a double fertilization. 2 sperm nuclei produced a diploid cell here as a haploid sperm nucleus fused with a haploid egg nucleus. And then here, the endosperm, which is now triploid, because 1 sperm nucleus fused with 2 polar nuclei. Thus, in a seed, it's rather complicated because this tissue is maternal tissue. It's diploid with only the maternal input. This is triploid, where 1/3 of the chromosomes are coming from the male parent. And then here's the egg, which is diploid with half of its chromosomes from the males and female parents, respectively. One of the reasons for this may be that a chromosomal abnormality in the triploid endosperm can cause to the failure of this, where the plant won't dedicate resources and the embryo will fail. And this may be a way of securing that only the pollen from the appropriate species fertilizes the egg. Otherwise, the plant might waste nutrients, helping an embryo develop that was in part parented by another species. So in this playlist, I've gone through the stages of plant life cycles, but you see, since the first plants made it on to land, maybe approaching 500 million years ago, to the first seed plants at the end of the Permian, or the first flowering plants in the middle of the Jurassic period, there have been shifts, and one of the most prominent shifts is the shift away from the gametophyte generation, which was once the dominant generation and the only photosynthetic generation, to, in the seed plants, the sporophyte being the dominant generation, which is the only photosynthetic generation.